It's a great pleasure to be back here in Lincoln. I see many familiar faces and some new folks as well. Though I'm being blamed for both the rain and the disappearance of the cookies. I'm not going to be invited back again. So today I'd like to tell you about this particular project, but lying behind this description of the project is a broader theme. And that has to do with a very strange notion. That is, creating courses that contain material that our students need to learn and not what we want to teach. That's a radical. So we'll let that percolate and we'll see how this plays out in this particular instance of courses for life science students. So I'm going to show you our agenda for the afternoon. I'll give you some introduction to this particular project, Living Physics Portal, just so you're aware of what it is, and then get on to the more meaty part of the presentation. I'll give you some context of why we're carrying out this kind of project, why it's an important one, and then I'll give you a series of examples where a physics point of view really helps students of the life sciences, and maybe even physicists and biology faculty as well, get some insight into how the principles of physics both enable and constrain what living systems can do. And they also, if framed properly, can help life science students understand better how living systems are taking advantage of these affordances provided by physics. And to show that this particular approach does benefit the students, I'll give you a bit of evidence from some of our research teams that are just beginning to come out of some particular instantiations of these introductory physics for the life science courses. That's the IPLS acronym floating there. And if we have time at the end, there's an issue which often comes up when talking about these kinds of courses for life science students. That is, for students who are interested in going to medical school, veterinary school, dentistry school, and so on, that require the medical college admissions test, the infamous MCAT, how is this going to subvert those students in their efforts in that direction? And of course, the answer is no, but I need to explain why, what that answer is all about. So this is a relatively complicated project. What we're trying to do is provide a platform in which faculty members who are going to be teaching these kinds of courses can find curriculum materials, pedagogical advice, course structures, the whole shebang that will be helpful in working with life science students. And we also want it to be a mechanism by which faculty who develop interesting materials in this area, and it turns out we're uncovering hundreds of such faculty across the country, it's a venue for them to share their materials with others, to get feedback on these materials, improve them, and hopefully this collection ultimately can be structured, and that's going to be the next stage of this project in the next couple of years, structured in such a way that somebody who's interested in teaching such a course can come to our site, say, here's how many students I'm going to have, it's this kind of course, most of the students are going to be in this particular field, they'll have this kind of background, what do I do? And the system will come up with some suggestions of people who have these kinds of students in this kind of course and use this package and this package. And these seem to work pretty well with these kinds of students, maybe not so well with these kinds of students, and be a much more sort of an expert guide for a faculty member who's trying to engage with these courses. As I said, it's a fairly complex project, so I have a lot of colleagues who are involved with this. There are more pictures there than there are people, because those people play several roles in the project. I have I'm employing about 20 people with NSF money, thank you, in this particular project. Most are physics faculty members, but we have some full-time web developers developing a platform for this. We have people who are working on research on the instructional content that goes into this platform and how to package that effectively. We have a group working on this community aspect where faculty can comment on other faculty members' materials and make use of things. a group that's handling the editorial processes. It's kind of a publishing firm in a sense. And then we have a group across the bottom of physics faculty who have been teaching this kind of course for many years, who are contributing their materials to get the project underway. There's another half dozen or so other faculty who fairly recently started contributing their materials as well. So it's a fairly complex operation, and we'll probably take another couple of years before we get this thing close to perfection. So why do we need this kind of project? Aren't there textbooks out there in this area, or courses where you just take your standard physics course and teach it to life science students? Yeah, it's physics, right? They should learn it. Okay. Well, first of all, for many physics departments across the country, this is a big service course. Over 200,000 students per year take this kind of course in some college or university somewhere in the country. And what makes this so complicated, as probably most of you know, is students come into that course from lots of different kinds of backgrounds, lots of different kinds of interests, and lots of different motivations. And you may have a lot of pre-meds, which a certain group to deal with. You may have a lot of students in the allied health professions. You may have hardcore basic biologists who want to go into biology research and genetics or some molecular biology. You may have ecology people, environmental science people. Extremely broad audience. How do you deal with that group? Complicating that, of course, is that most of us have not grown up with a whole lot of background in the life sciences beyond, last time I had biology, like to joke, well, maybe it's a joke, this is before we knew about DNA. We knew about DNA, but we didn't know about it the way we know about it today. So most of us don't have much background in this area, so how do we figure out how to teach those kind of physics things that are related to biological topics? And I said textbooks, but really if you look at the textbooks which are out there, most of them are totally inappropriate. They're really sort of an algebra-based physics thing with some pictures of tigers and things like that, and really not real life science applications. That's changing. There's some new books already out there. More recently, there's several in the pipeline that may change. And what makes this all much more complicated is the life sciences themselves are changing dramatically, both at the basic research level. In some ways, they're becoming a lot more like physics. There's a lot more quantitative analysis, a lot more computational modeling, some of which is going on here at UNL. A lot more mechanistic thinking about how genes do various kinds of things and what those molecules are doing moving around inside cells. So it's beginning to look a lot more like a physics perspective. And most biologists admit they're not quite sure how to wrap their heads around that. But their students sense that this is coming. And so that makes it the subject matter even more challenging to deal with. Also makes it very exciting. Okay. So I think I've sort of hinted about why you might want to do this course differently from a standard physics course. But from a fundamental learning perspective, we all know, we've all experienced this ourselves. If you learn something in one particular context, it's difficult to take that and move it over to a new context and apply it. We've experienced that in our math courses, for example. You learn all about partial differential equations in the math department, and you come over to physics and, how do you make this stuff work? But it's equally true for, say, physics content. Students may understand all the basic physics principles and conservation laws and things of that sort. But then how do you apply this to this complex system in biology where there are many different competing length scales and time scales? Learning to make that transfer is difficult. And so allowing the students to have opportunities to do this right in the class will be wonderful for everybody. And there's also the inspiration, the motivation question. I think students are much more motivated to do the work of learning the material if they can see how it's going to apply to their own professional interests. That's just the way we humans are. It's also good for physics in a sense because you can be better at selling physics to your colleagues on campus or the dean or the provost because you can say, we are helping all of these students across campus further their professional careers in a way that only physics can contribute in that particular area. And of course, from the student's point of view, what they need to know about basic science, either in biology or physics, probably they can't guess, because what's going to happen in biology in 10 or 20 years, we just can't imagine what's going to be like. So you really need a firm grounding in basic general principles, which is what physics is really good at. And we can take those principles and, as fields evolve, apply them to new circumstances as they arise. So, product right now, our portal is a website, which is just a beta version. You can come and poke around a little bit. We don't have all the stuff ready to go completely live. But you can see from just this screenshot what the basic things are. There are curricular materials, which you can search in a variety of guided ways. And they come in a variety of forms. There's what we call a community library, where anybody can stick something up, at least as long as it has something to do with physics, with life sciences. And they say this may not be polished, they just had this great idea over the weekend, see if you think about it. Some are things that have been used in some classes, but not highly tested. Then we have a vetted library, which is a little more sophisticated material that's been used a number of times, some evidence about its effectiveness for different audiences. Our editorial team goes through it, make sure it's clear and coherent. It's not quite like detailed copy editing for publishing, but we make sure it's pretty good stuff. Then eventually we do intend to have a detailed peer-reviewed section, which will be more like an online journal where things will be checked out much more carefully. That will come in the future. We have a community section, as I mentioned before, where people can comment on things that are on the portal and leave some suggestions and comments. They can also say, gee, I'd like to find some materials on topic X. I don't see it there right now. And perhaps somebody who's associated with the portal will say, oh, I've got some stuff, and provide that as well. And then there will be a contributing section where you can come in and add your material and get feedback from that. And I hope eventually this will become enough of a peer-reviewed platform that will be some professional recognition about your contributions here. It's not just random thoughts. It's been vetted, checked, and deemed to be fairly trustworthy. So again, when you do the search, you can search by physics topics, biology topics, the type of pedagogy that's used, whether it's being used in lectures, lab, or homework, or any other mechanism. And as I mentioned before, commenting on the materials is going to be an important part of the process. So what also motivates us besides those pedagogical aspects I mentioned before? Biologists have been thinking the last 20 years or so pretty deeply about where their field is going and what it's going to look like in the future. There is a report called Bio 2010, 2010 has come and gone, but it really points out in that report how biologists think about the field and its evolution. And they mentioned the things I mentioned before. It's becoming more quantitative, it's more mechanistic, it's more relying on detailed, quantitative, theoretical studies of things than biology has been in the past. And that's just the way the field is going, and you either hop on the bandwagon or you don't. And of course, we're encouraging biologists to help prepare students in these directions, but they also recognize that in mathematics, in chemistry, and physics, all those disciplines can also help prepare students for what biology is going to be like. Another report came out about 10 years ago called Scientific Foundations for Future Physicians. The title sort of tells you what that was all about. I was part of that particular project, which looked at both undergraduate and medical school basic science instruction for future physicians. And it was really quite an enlightening enterprise because we had to look very carefully at undergraduate science and math education and really think carefully about how that was feeding into preparing future physicians. And then the medical school folks really did a very rather interesting analysis of the basic science that medical school students take. And that was eye-opening for me to realize, and when you look at the list of stuff, 90% of it is physics-related stuff. So if you need to convince your students that taking this kind of course is important, point to that document. It really lays it out very carefully. Then there's also more recently a document called Vision and Change in Undergraduate Biology Education. I wanted to take a more principled view of biology education. It's not just a collection of a dozen different courses on different topics about plants and animals and cells and genetics and stuff. They tried to think about the general principles that interlock all of these various approaches to biology, and I think came up with a very nice formulation. A lot of it involves, of course, fundamental things like evolution in biology. That controls everything. But also thinking in broader terms, quantitatively, models, theories, evidence, all that, which, of course, in the traditional physics bailiwick has not traditionally been part of biology education. So that report lays that out very nicely and feeds in with all these others, all pointing more or less in the same direction. And all of these reports, it's also emphasized that not only should you worry about your content in your courses, you should also worry about how you teach this and engage students in active ways in that education. And this project really got rolling a few years ago. We had a conference which brought together somewhere in the order of 150 or so physics faculty with some biologists to talk about what these kinds of courses would look like and try to put together a report which outlined the kinds of things that physics faculty were looking for in terms of materials, pedagogies, and so on, to help make all of this work. And things, fairly obvious things like how do you do labs in for this kind of course? How do you make this a physics course? It still is a physics course, but one that is aimed at life science students. And everybody realized one of the big problems is when you have these multitude of different kinds of audiences in different institutions in these courses, you can't just have a How They Resonate Physics book or its clones, as we do in physics. You have to have things that are tailored, or curriculum materials that are tailored to your audience, for your students, their backgrounds, and their professional interests. So building more textbooks is not going to be the way to do it. That's part of the motivation behind this portal project, where you can assemble materials. Biologists themselves have weighed in on this. And here's a quote from a biologist who wrote a really nice quote about the biology and physics of airborne. That's where living systems as we know them live. And that's the physics behind all of that. And again, pointing out this general notion that physics both enables and constrains what living systems can do. Materials of this sort also become available. It probably is not suitable for a textbook or an intro course, but it does give you the physics faculty members some background, or students who are particularly interested in pursuing some of these problems. There's also the technology side of things. Students get to medical school, they got ultrasound, they got MRIs, they got functional MRIs, all these single cell microscopy techniques, single molecule microscopy techniques that they use nowadays to understand living systems. It'd be nice if they had at least some idea how these things work and what their limitations are. And so again, that could be a theme or a sub-theme in a course for a physics or life science students, depending on how you want to proceed with those. So lots of connections here that we want to build upon. Of course, this focus on physics for life sciences has a long history to it, going back many, many decades. In fact, Bob Fuller and Vicki Clark, who were here at UNL, had this Humanized Physics project from several decades ago, which produced lots of interesting materials, which are still floating around, some of which are being re-massaged into our Living Physics portal. Big group at the University of Maryland has been working on this for many years. At MIT, for at least 30 years, at Lars. Some very nice, pretty sophisticated books for physics for life science students. And those I found not so good for students, but again, good for faculty members to get some ideas of how you take powerful physics ideas, statistical mechanics and E&M and everything else, and apply that to living systems. And then you can extract that and massage it appropriately for your particular students. So it's not as if this is a brand new idea, but we're trying to approach it from a much more flexible point of view, and also a little more sophisticated point of view in thinking about the pedagogy that goes along with this. So what I'd like to do is give you now a quick run through of a couple of examples of kinds of things that might be integrated into this kind of physics course for life science students. And the language we've been using as part of the project, we call these authentic examples and problems, where authentic means that the physics treatment of these things helps you understand and at a deeper level the living system that you're taking a look at. Now the ones I'm going to show you, biologists say, well, that's not very deep. And maybe some of the physics is not very deep. But think of this in terms of a life science student taking Steve's course, for example. And you're trying to show them, teach them some fundamental physics, but along the way you want to give them a glimpse of how that physics helps them understand living systems. So you want to pick a good living system example where relatively straightforward ideas in physics help you understand what's going on. So I'll give you a couple of examples. Some are pretty simple. One of them leads into some very new and active research on cancer, all done with introductory physics kind of stuff. So here we are. The first one is pretty straightforward in some ways, but something you may not have thought about. This has to do with birds who stand often in relatively cold water, but they're warm-blooded animals. How do they negotiate those temperature differentials? Well, those birds are very clever. They build up a heat exchanger, nature provided it for them, where, of course, you want your feet are going to be cold, so you don't want to lose a lot of body heat to that environment. So you have to circulate blood through there to get nutrients to your feet. So you cool the blood down, it goes to your feet, by having it interact with the blood. And you can just point out that all along that contact point, the blue stuff is warmer than the pink stuff, and therefore thermal energy will flow in that direction. Very simple principle, but engineering it that way, so you have contact over some distance, nature was pretty clever about that. Okay, next example. This may seem like a crazy one, but a biologist working with physicist Joe Reddish at the University of Maryland actually raised this question when they built this into their course. We know worms are around us all over the place, but worms are always more or less the same size, a couple millimeters across, a couple centimeters, or maybe a little bit longer. And the biologist asked, why don't we see giant-sized worms? Well, if you see the movie Dune, remember there's this thing that goes through the sand, which is a big worm. But outside the movies, you don't see giant worms going around your backyard. And the biologist, who's actually a botanist, said, well, I know that worms don't have lungs, but they use oxygen. So how do they get the oxygen from? Oxygen diffuses through their skin into the body. And so the biologist said, how does that information understand what constraints nature puts on the size of the worm? So if you think about that, like physicists would think about that, they would say the oxygen needs go up with the number of cells in the body, the volume of them. And if you imagine you have a cylindrical worm, goes along with spherical cows and the physics, right? Cylindrical, it's not a terrible level. And so the volume is the cross-sectional area times the length, approximately. So it goes up as the square of the radius of the worm. Now, if oxygen is diffusing in, it comes through the surface. So this is an area effect. How's the area of the cylindrical worm? Well, it's the circumference of the worm times its length. Now, as the worm starts getting bigger, fatter, this radius is going to increase. You can see the volume requirements go up faster than the surface area. So this is limited. There's some diffusion factors in there, but you're not going to change a lot. But basically, that tells you if the worm gets too big, it can't get enough oxygen in, so it's not going to get big. So that fact that it's getting oxygen through diffusion is limit, and it's coming through the surface area of the worm. That's what limits the size of the worm. I can put real numbers to this, but that's the basic qualitative idea. And of course, that's, for a physicist, that's a standard way of thinking about these kinds of issues. You may not talk about worms, but you can think about similar things in other kinds of materials. But for biologists, that's not a typical kind of reasoning that takes place. So this is a way that the physics kind of reasoning helps understand that biological question. Coming back to diffusion for my third example, if you ask biologists, biochemists, and so on about what's the one physics topic you wish the students would really get in their physics courses, it's diffusion. Everything in living, not everything, lots of stuff in living systems depends on diffusion, right? There's lots of stuff inside a cell. It's got to move around, get here and there. And things have to diffuse from blood vessels into muscles and from nerve cells to all stuff. It's all driven by, a lot of it's driven by diffusion, not everything. Quite a bit is. So how would a physicist approach diffusion? Well, we all know this stuff. Well, maybe you can remember it vaguely. And there's a thing called Fick's law, which tells how diffusion rates are related to concentration differences, and flow depends on the gradient of the concentration, it's a coefficient, and then you say, well, this needs to be spread out in space, so you have spatial dependence as well as time dependence, and so you end up with a partial differential equation, at which point you've lost all of your life science students. Right? What do you do with this thing? Well, it's from a physicist point of view, or maybe a junior physics major, it's not too hard to deal with that kind of stuff. But it's really not illuminating very much because it gobbles up all the microscopic stuff that's going on in diffusion and hides it under an average concentration and a gradient and all that sort of stuff. So what could you do to make that a little less mysterious? And I think from a pedagogical point of view, here, I've tried this over 20 years with different kinds of students, and it works like a charm. Better is to start with a more microscopic modeling of diffusion. Now, physicists always have to have collisions and everything. You can make it even simpler than that. And I first learned about this particular way of doing things when I was reading some history of statistical mechanics. Back in 1907, Paul and Tatjana at Ehrenfest decided they needed to write a book on statistical mechanics, which was just emerging as a science field at that time. And they wanted some simple model to illustrate some basic principles. So their model, they used balls and urns, but I think it's more colorful to use dogs and fleas. And so the situation you imagine is that you have two dogs, Lefty, they're just outside playing with a neighborhood cat, picked up a whole bunch of fleas, comes back in and sits down next to Righty, and the fleas, being fleas, start jumping from dog to dog, because that's what fleas like to do. And so you can say that, the fleas are not very coordinated, so they're just going to jump randomly at random times back and forth between the dogs. And the probabilities of going left to right or right to left would be equal, at least in the simplest version of this kind of thing. So then you ask what happens over time to the distribution of fleas. They all jump over to the right, or they all jump back to the left, or what happens in between. And it turns out it's a very simple task to make a little simulation of this. I'll show you the results of the simulation. It turned out the network here is not very happy with this URL, which links to the actual program itself. But the program itself is maybe about 20 lines of code, 18 of which make the picture. And the other part is this random number saying it throws a random number, and that numbered flea is on the left and it jumps to the right, and that numbered flea is on the right, it jumps to the left. Very simple. And wouldn't expect your students to do the coding, but they're going to use this. So then you ask the students, well, suppose we want to plot the number of fleas on the left minus the number of on the right, sort of the population difference. What's going to happen as jumps start occurring? So in this picture, the upper sort of turquoise, greenish kind of colored area on the left shows you your cylindrical dog with fleas on it. And then you can see when I grabbed a screenshot, a few fleas had jumped over to the right. And I have a little graph here at the bottom. The blue line is going to be the population difference. And I had 100 fleas, so it starts at 100 fleas on the left, not on the right. And you can see that we've got a few fleas off to the right, so that came down. So the question is, what happens in the long term? So you can think about that for a second. We won't ask for hands or quicker questions or anything like that. We don't want to be embarrassed. But you think about what's going to happen. And of course, if somebody's taught statistical mechanics, of course, they know what's going to happen. So this shows a longer term run of this. It's had 900 jumps going on here. And you can see the sort of bluish line at the bottom of the graph is the population difference. And as expected, perhaps if you think about this, it drops down close to 0, but it bounces around. For statistical fluctuations, I can say those words, these things bouncing back and forth. The red line is proportional to the entropy of this situation. For the physicists, it's logarithm of the number of available states. And you can see the entropy goes up. It's an isolated system working itself toward equilibrium. We all know the words to say. And you can see it goes up. But it also is not absolutely flat. It wiggles a little bit. And any finite-sized system like this, you're going to have those kinds of fluctuations. That turns out to be a revelation for most students. Because they hear about diffusion in the biology, somebody draws a graph, the solution of that partial differential. It comes down exponentially, hits 0, and it's just flat, forever. So from the student's point of view, equilibrium is that when the population difference is 0. And at that point, everything stops. Those wiggling things that were molecules, supposedly, might be jumping around, they all stop moving. And it's just quiet from there on. In real life, we always have fluctuations. So that's a really important lesson, because those fluctuations in living systems are absolutely crucial to make the living system work. There are even fluctuation-driven oscillations and things like that in biological systems. So this is really a sock-at-home message, and it just opens eyes tremendously. So the lesson you learn from this, or hopefully your students learn too, is that, first of all, say, how do fleas know to jump this way? Well, they don't know. Fleas, like electrons, are pretty dumb objects. Okay? All they know is, I'm going to move around randomly, get from one place to another. But statistically, it drives the system to lower population difference. So the fleas don't have to know anything to make this happen. And there are fluctuations along the way to equilibrium, and there are fluctuations when you are at equilibrium. Equilibrium is a dynamic state. That's really the take-home message from all of this. And so what this also emphasizes is that many situations in biology, this kind of microscopic statistical approach is really the key to understanding what's going on. And in a typical cell, so we won't go into any detailed biology here, but suppose you're tracking certain proteins, slipped into the physics mode, proteins in a cell. There might be only 10, 20 copies of a particular protein inside a given cell. And so that protein might be moving around from place to place, but there are going to be tremendous fluctuations in the availability of that protein to be at the right place through the do the right biological function at the right time. Lots of important lessons from this. And this model also can be applied in other situations. Here we talk about fleas, but you could say this could be molecules moving around from one place to another. It could be across a membrane. It could be osmosis-like kinds of things if you add a little some pressure mechanism into this. It's also a model for heat flow, thermal energy going little packets from one place to another. Let's start on the left. Lots of thermal energy. It's hot. Little thermal energy on the right. It's cold. On average, they always move from hot to cold. So there are lots of ways you can play this out. And I think you can build a whole statistical mechanics course around this. I mean, it's extensions. So, moving on to yet another example. Okay. This one has to do with cell membranes and the electrical voltages associated with cell membranes. So this gets into electricity and magnetism for physics course. And again, in our 10-second review of biology, a typical cell may be a micron, 10 microns across, but it's bounded on the outside, and there are some boundaries on the inside, which are very thin membranes. And those are really important in compartmentalizing and distinguishing things outside the cell from inside the cell, or in the center of the so-called nucleus, that's where most of the DNA, stuff like that, is separating that from the rest of the cell machinery. So these membranes are absolutely critical features of almost all living systems. And in most cases, there is associated with those membranes a electrical voltage difference between one side and the other. So on the outside boundary of a cell, there's the insects being blown up. The membrane itself is sort of a layered structure here, something in the order of 5 to 10 nanometers in thickness, depending on the particular cell. Now, they're not really just homogeneous. There are a bunch of stuff stuck into those membranes. There are proteins that go across here, things that make channels, there's stuff on the surface, there's stuff on the inside. So it's a pretty complicated system, but that's the way our cells operate. And in most cells, there's an electrical voltage difference between one side and the other. It's typically on the order of 1/10 of a volt, 1/10 of what your 1/10 volt battery C cell can make. And typically, in many cells, the outside is positive with respect to the inside. There's some exceptions to that. Now, for the physicists in the audience, do the following little mental calculation. You know, if you take the voltage and divide by the distance, that gives you an electric field. That's a big electric field. 10 to the 7th volts per meter. Don't curl your hair, right? If you have it. The air breaks down, about 50,000, 10 to the 4th, something like that, 10 to the 5th. So this is a lot larger than that. So that means that ions that are in the vicinity of this cell membrane, on average, can experience very strong electrical forces. Fortunately, we don't get too many sparks in our cells, but yeah, some other materials there to help avoid that particular issue. Okay, so you can help the students understand the stuff. They probably heard this in their biology courses. And they always say, it's positive on the outside, negative on the inside. But, some orders of magnitude numbers really help make that more concrete. Or sometimes the biologists will just talk about it in terms, well, it's lower voltage on the inside, period. And students have a tough time sorting this out with the way we talk about voltages in terms of higher voltage, lower voltage, things of that sort. So you can help them work their way through that a little bit, which is helpful to them. And you can also ask questions that biologists typically don't ask. For example, you say, here we have this membrane, this 1/10 of a volt difference. Imagine we have a surface here. We know that one side is positive with respect to the other. So the simple way of thinking of it is we have positive ions on the outside, negative ions on the inside. maintain that tenth of a volt, you have to have lots of ions packed in, or just a few. Okay. Physicists know what to do. Travel plate capacitor, charge density. You don't have to do the calculation right now. But I was looking a little nervous there. But easy enough to carry out. And you find out, and assume, just for the sake of argument, that these ions are spread uniformly across this. And it turns out the typical distance between the ions is about 14 nanometers, more or less same order of magnitude as the thickness of these things. Maybe makes some sense. Draw some pictures of that. Another way of thinking about it is think about how big is that distance compared to the size of an ion that might be out there. Most ions, of course, in cells are like a calcium ion that's going to have water molecules all grouped around it. But think of the calcium ion itself. And you see that it turns out it's going to be about 100 times the size of a typical ion. So if you imagine yourself 1 foot across, well, maybe it's not a little bigger, it's years ago, but roughly a foot across, the next ion is 140 feet, half a football field, the basic Nebraska unit of measurement, down that way. So those ions are pretty far apart. They don't interact with each other directly very much. That's important for many biological processes. These things float around more or less independently of each other. Okay. You can also extend this a little bit. There's a long history of doing this. You can make circuit models for these cells. And here is one particular version of it, the cell membrane, we treat as a capacitor, those parallel layers. You have a voltage, which here we mimic it. We'll show it in a battery, because it's not a real battery inside your cells, but there are mechanisms that produce that voltage difference. And then there's some resistance or conductance, depending on how you want to talk about it, across that membrane. I showed you all those things that stick through the membrane. So some ions can get in, and some ions can get out. And it turns out many of those channels for the ions to go in and out are determined by the voltage across the membrane. So these are voltage-controlled systems. I'll show you how that plays out in a second. But you can make a diagram like that. You can even say, if I change the conductance for sodium ions, what happens, and so on and so forth. Particular application I will look at has to do with nerve cells, which are long, thin cells. And they carry electrical voltage pulses. That's how we move and think and all those good things. And for a long time, people have done this kind of modeling of these long, thin nerve cells, treating them as electrical circuits. So you have capacitance, the letter C there. You have resistance across there. Inside the cell, it's a kind of constricted region, so the resistance is moderately high. Outside the cell, lots of water and ions floating around, so the resistance is relatively low. And you can use that to say, well, suppose we jiggle something over here on this side of the cell. What happens to that change? How does it propagate along our electrical cable? And it turns out, of course, all physicists say, well, we have resistors and capacitors. It takes some time for this to readjust itself. Typical characteristic time is given by the resistance times capacitance, and we've learned something from that. Okay. So, now, the particular applications interesting are, as I mentioned, are nerve cells. And these nerve cells, that long, thin thing up there in the middle of the top diagrams, that long part is called an axon. And the simulation that's running there shows you the typical conditions, that the plus charges on the outside, minus charges in the middle. When a nerve signal travels down there, those charges swap. And so you go from being positive on the outside, negative on the inside, to the reverse, negative on the outside, positive on the inside, and that, because things are voltage gated, that propagates down this nerve axon. And this has been known for a long time. In fact, people who are related to physics help do some of the first measurements to determine some of these kinds of properties. And the diagram on the bottom shows a typical nerve impulse. The voltage starts off down here. You know, the typical inside is negative with respect to the outside. This has a 0.07 volts, close enough to 0.1. And then when the nerve impulse fires, the transmembrane voltage goes off into the other side. Sometimes biologists say this is depolarized, and then there's a recovery here, and then you end up back at this negative level again. And all this happens in a couple of milliseconds. And that's a typical nerve impulse. And up here at the top, this has been annotated with some information about how different ions behave under these circumstances. When this nerve impulse starts, sodium channels open up. There's higher concentrations of sodium outside the cell and inside, so the sodium ions flow in, and that's going to reduce the voltage difference. And that goes up to the peak, and then the sodium channels shut off. Potassium channels open, there's more potassium inside than outside, it flows out and reestablishes that negative voltage after the fact. And in fact, it overshoots a little bit, which turns out to be very important. This overshoot here means that the next nerve impulse can't come until this thing is completely recovered. And so you can't have nerves firing too quickly, maybe up to a couple hundred times per second. Otherwise, there's not enough time for this thing to recover. And that limits all sorts of sensory things, muscular things, all because of the physics of that membrane potential. So you can talk your students through this kind of thing. Probably those who have taken a neuroscience course probably have heard some of this stuff. They haven't really sort of gone through step by step. Which charges move where, why does that make it positive, why does that make it negative? So you can help out with that kind of stuff. As I mentioned, lots of physics folks were involved with this. There's a famous Hodgkin-Huxley model of nerve conduction developed back in the 1950s or so. Hodgkin and Huxley both worked on radar stuff for World War II. They had strong physics background, came out knowing how to make oscilloscopes and other electronic information work, and they decided to apply it to nerve cells and carried out some of the first detailed measurements of these so-called action potentials in nerve impulses. Now, a nice physics question, or physics question you pose your students. Many nerve axons, long fibers trailing off, make contact with other nerves, particularly in the peripheral nervous system outside the brain, have a little coating on them, a myelin sheath, sort of like heat shrink, wrap around the plastic, wrap around a piece of wire. What does that do for me? Well, from a physics point of view, we know that it's an insulating material. So any of the ions that are present are going to be outside that. So now, instead of having just a membrane thickness determining the capacitance, you have this thicker layer. What happens to the capacitance? Or is that like that? All right. For a given voltage difference. Sorry. Yeah. So anyway, capacitance goes down. What happens to our RC time constant? It goes down. So it can respond faster. And the peripheral nervous system, that's what you want. You want those nerve impulses from your brain to get out to move your arms and legs more quickly. And that turns out to be a very important mechanism. And when that myelin sheath is injured, or chemical things or even physiological impact things, and the nerve impulses don't travel far from this, and it's not a nice state of affairs. But that's a nice example for students because it ties the physics, the decrease in the capacitance, the faster impulse travel time, which is what you want biologically. But then you can see why damaging that is going to mess up those nerve impulses in the peripheral nervous system. And it helps you understand why that particular functionality is particular and motor performance. Okay, and this also happens to tie in. I see we're getting short on time, so I will move right along. Another interesting aspect of this membrane voltage difference is that as cells go through a cycle, a cell is born and constructed, it lives for a while, and starts to grow, and it decides it's too big and it's got to split into parts. So it duplicates the DNA in the cell, and then the cell splits apart. While that's going on, the membrane voltage is changing from the normal negative values, in this particular example, minus 40 millivolts. You can see as it moves up toward the place where the cell starts to grow, that voltage decreases in magnitude. And then when it's starting to grow, it becomes more negative, and then when it's ready to split apart, it goes back and it tends toward positive ranges. And so that's all tied together. It's not, my understanding is that the details of exactly how that works is not completely understood yet. But something's going on with the membrane that signals the stuff inside in some way that causes these changes in voltage, but it tracks very nicely the cell cycle. Now, there's some cells where the cell cycle goes crazy, like cancer. And it turns out that cancer cells have a less negative membrane voltage than other typical cells. Across the board, almost every type of cancer has that. What does that have to do with it? The thought is that somehow it's screwing up the cell cycle, and cells are just growing and growing and mutating and splitting, and things are not controlled. They don't get back to the sort of steady state value. But and somehow that cell membrane voltage is tied into that. Whether it's a symptom or a cause, much is known. More current research, a September issue of Science magazine, an article about how nerve cells, friends, might be in cahoots with cancer cells. Which sounds absolutely crazy. It seems there's some evidence that this happens. And it turns out these cancer cells, are the gray globular things here, are aided in some of their bad behavior by what nerve cells can do. For some reason, nerve cells growing out, these finger-like things, are attracted toward the tumors. Exactly what's going on in the nerve cells up there. It happens. And the nerve cells can release some chemicals in the neighborhood of the tumor, which encourages the tumor to grow. Some of the nerve cells can scare off the cleanup crew from your body, which can attack cancer cells, pushes them away. The nerve cells can stimulate blood vessels and provide more blood for the tumor. And the thought discussed in this article, maybe it has something to do with these membrane voltages. But I'm giving this example, it's not something you can actually maybe do in class in a formal way, but it's a way of letting students know that some very simple ideas like this membrane voltage, which has a lot of physics in it, carries over to some frontier research dealing with serious stuff in cancer. And if we knew more about what these membrane voltages were doing with cells and their behavior, we might make some progress in learning to turn off some of these other mechanisms. I think that's another motivation for students to take a look at these things. Okay, so I see I got too excited about some of these physics things, so I'm going to skip over this particular one, except to warn you, of course, in physics we love to talk about energy. You say, oh, that's good because we know biologists talk about energy, bond energy, and all that kind of stuff. Turns out we're talking two different languages almost entirely, and students need some help unpacking that kind of thing, because biologists like to say energy is stored in these strong chemical bonds. Physicists, molecular physicists, know it takes energy to break bonds. It's stored in it. So there's a lot of context you have to unpack to make these kinds of things work. Physicists can help understand it. So skip it over. let me get to the bottom line about whether all this work to restructure a course for life sciences students is worth the effort, because it does take some effort. One study our group has been carrying out is at Swarthmore College, where they've been implementing this introductory physics for the life science course for a couple of years now, so they have a cohort of students to follow along. And they found that two interesting things are happening. First of all, the students report that they liked this course because it gave a kind of coherence to their study of physics. It gave them a framework on which to hang all these various physics ideas. Of course, developing that kind of intellectual framework is really important in really learning something to the extent that you can use it under real life situations. The other thing that students mentioned quite frequently was the fact that many of these examples dealt with human body kinds of situations. And that gave them, that's important to me. I want to figure out how all these kinds of things work. And at Swarthmore, they have a little experiment where they do simple electrocardiograms, just some simple electrodes based on their arm. And just being able to see that, they hear it, they've read it in the textbooks and heard their doctors talk about it. Actually see, you can measure that, and you can see things happening in your own body. That's really a powerful kind of message. And the project is going on long enough there at Swarthmore. They see this way of thinking persist with the students as they go on to upper level biology courses. Professors recognize the different way students have talking about the biology. One other case study, and then quick, and that is Rutgers College, which is a Jesuit college in Kansas City, about 2,300 undergraduates. Historically, they have a lot of pre-med and allied health students. That's their shtick. They get students in. And one of the physics faculty members, Nancy Donaldson, see her pointing there at some graphs, decided, well, if we have all these students who are interested in pre-med and health sciences, why don't we create a program which really takes that interest and blends it into physics. So she developed a bachelor's degree program in physics with a physics of medicine concentration. She went from 1 or 2 majors per year to about 20 per year. Thank you. And those students are getting into medical schools, are getting great jobs in the Kansas City area, working with medical firms and technology firms. So high quality stuff. And it's all, this is sort of building this theme in of how to think about physics as applied to these particular topics that are of interest to the pre-med and the allied health professions. And that's an example of where you can really turn around a physics program by recognizing the needs of your particular students. That may obviously not apply in every institution, but I think there's a general principle there. Know what your students are interested in doing, and then build a course that helps them further their professional careers. So let me stop there, and thank you for your attention. I'm happy to take questions. Yes, sir.
Yeah, does living physics model is still in beta?
Yes.
Do we have a timeline on when we'll be live?
Smile. We hope in a month or two.
Oh, okay.
Yeah, we're getting close. There were just some things about, in some sense, trivial things, but things you want to have working. Like being able to have people leave comments in the right place, and the authors get an email message. Crazy kinds of IT stuff, but it takes a long time to get all the ingredients working properly. There were some glitches in the editorial side where people were submitting things, and reviews were coming back, and just making sure everybody gets notified. So we're getting pretty close. And then ultimately, as I mentioned, we really would like to have this course builder model implemented, and that'll be another year and a half to implement that. Let me take one back here, and I'll come back to you, Tate. Yes.
So I actually TA for a physics lab that's geared towards predominantly life science majors.
Right.
And that model you showed where modeling the action potential of a neuron, we actually do that using a simple RC search. And what I'm wondering about is essentially kind of limitation of instructor knowledge, where I could talk their ears off about, here's an RC circuit, here's the time constant, here's how it behaves. But as soon as it came to translating that to its application to this biological aspect, right? I haven't had biology since freshman year of high school, so I could do very little. And I guess sort of what are your recommendations for instructors, especially in institutions that haven't gone all the way into this sort of program yet?
Sure, absolutely. I would suggest the following. This is my sort of bumper sticker phrase of advice. Take biologists to lunch.
I don't have money for that.
How the biologists take you to lunch? Find a biologist that you could talk to and is willing to spend a little time with you, say, and do it piecewise. You know, I want to say take the action potential one. I want to say a little bit more about that. And of course, you can do some background reading yourself. You know, lots of good books, some written by physicists that help you along in that direction. And then talk to the biologists. How do the biologists talk about that in their classes? Where's Brian? There he is. Look, Brian.
Can I just say that not all biologists know all of biology? So
Well, that's why I said it's plural, biologists. And so depending on the topic you're interested in, you would want to meet up with different biologists to get their perspective and say, what do you wish your students knew about the physics side of this coming into the courses? What are good ways of thinking about this? What's the language the biologists use? So you can do that kind of impedance matching with that. And some of it just takes time over. You do some reading, some good books, as I mentioned, about this kind of stuff that can help you get up to speed. You're not going to be an expert, but you don't need to be an expert. You know the basic background. And as a physicist, you can sort of sense where the physicsy part is, and then like these nerve axons, there are 20-some different potassium channels, something like that, which have different characters. You don't need that stuff. So you have to have some sense of balance of what level of detail you want to get to.
Okay, thank you.
Tim.
You did a nice job of showing why worms can't get too big. Why can't they get freakishly long?
Well, that was one of the bottom of the slide. That's sort of an extension question. Why doesn't that happen?
Yeah.
And so you really want to think about worms, you know, as they move. There's obviously some correlation, coordination of the various segments they can wiggle along. I'm speculating, I don't know. But we speculate and then talk to some wormologists. It probably has to do with being able to coordinate the various kinds of segments as they move along. Their nervous systems are not very sophisticated. Whereas a snake has a pretty sophisticated nervous system. I think a 30-foot snake is no problem at all.
Yes.
So I mean, that kind of question is the great kind of question to raise because it says, oh, there's more of this than we just looked at. How would we think about that? What kinds of questions would we want to ask? Where would we get evidence? What kind of mechanisms would we look for? That's a superb kind of thing to do with the students. It gets them into that questioning mode. They see things, obvious questions are sitting right there.
So let's say, you've got a standard course, so and, you want to do this, but it's totally revamping a course is a lot.
Absolutely.
How would you recommend getting started?
Well, start small, right? With many big projects, right? You start small. And I would suggest, for example, looking at the Living Physics portal, just with lots of different topics they have there, and say, what are one or two things that I might work into the course, just to see how it goes. I talked with people like Steve and Heath. Sorry. Terrible names on first sight. So who've been doing a little bit of this, see where they got started. And of course, on the portal, we're eventually going to have lots of faculty advice. How do you get started? Where are some good entry points that aren't too hard for you, aren't too hard for the students? And sort of build up your repertoire over time. Good. I don't think that there's any other way to do it. My own history of this is a similar thing. One of the first courses I ever taught as a professional physicist was Introductory Physics for Life Sciences students. So I put one or two things in, and then students said, oh, that's cool. Why don't you offer, we had at that point, a winter term, or these little mini courses that could be constructed very informally. Students said, why don't you do a mini course on this and that? I said, well, I know nothing about it, but I'll sit down with you guys, and we'll figure something out. So over a period of a decade, I gradually built up my repertoire of things like that. It does take time. And so I think it's perfectly fine. Start small, work with what's comfortable for you, find out what engages the students most readily, things like that. Yes?
So I'm curious how you plan on spending that time in building them up. So like in the case for electrostatics, so will you spend a bunch of time teaching them Coulomb's law and charges and then try to bring in more complicated scenarios from biology? Is that the idea?
Yeah, well, I would say in this particular situation, you need the machinery to get to know what parallel surfaces of charges do with each other. There are lots of different ways of getting at that. So if you can get that far, then that's far enough to do all this stuff with cell membranes.
So I just
In
meant,
fact, Coulomb's law sort of fades into the background.
right. I just meant ultimately it'll be more material, so you'd have to cut something out, right? So you maybe have to leave something out.
Yeah, you don't do
I'm
much
not saying you shouldn't, I'm
don't
just
do
saying.
much with planetary motion. Kepler's laws,
Yeah.
one of my favorites. It's painful. But and you can't, even with a regular course, you don't do everything. And you make selections of some form or another. So this is just another selection. And as long as it benefits the students, and it's a selection worth being made. So coverage is not a big deal. I had promised to say something about the MCAT, but we ran out of time. But the MCAT is a minor, well, first of all, a minor thing that medical schools use in admissions. It's not a big deal as soon as seen. And if they say explicitly, covers a very limited number of topics. You're not going to need, you'll need a lot more if you're going to be successful in medical school, both in the biological side, the chemistry, and the physics side. And the stuff that's on, the physics stuff that's on the exam would be covered in any kind of physics course, unless it's all general relativity or something like that. But any typical introductory course, you're going to take care of the coverage the students need to get that over that little barrier. And so I wouldn't worry too much about it. There's lots of good stuff you can do, and maybe a little bit different from your emphasis in a physics course for engineers, for example. But it's still all good stuff. Physics, all the basic principles. Anything else?
I have a quick one.
Oh, Keith.
All of your examples are mostly E&M based, but I have a terrible time finding ways to relate kinematics to biology. Can you give a quick example of a substitute kinematics example in biology? Something other than like the Arctic fish firing water at a bug.
Yes. Well, there are certainly lots of mechanics things at the level of large body motion, torques and forces,
Sure, right.
joints and so on.
Yeah, but like.
And I think in terms of understanding living systems, projectile motion, yeah, I mean, it's important for those fish. They should somehow have figured this out without doing too many calculations. So, and it's kind of interesting to see it happen, but not so critical. So I would say there are lots of things in mechanics you just park off on the side. You don't, in many of these introductory physics courses for life sciences, there's much subdued attention paid to kinematics and constant acceleration, none of which ever happened for living systems anyway. Right? Everything's viscous, movement, inertial. Right? So you operate in that realm. And as long as you know, the students know a little bit about speed and things like that, that's fun.
So the answer is do less.
Pardon?
So the answer is do less.
Do less of that. That's a place where you give up a little bit of stuff. And then that opens up a little bit of time to do something else that would be more comparable and illuminating for life science systems.
Let's thank our speakers.